Dear students, good morning. Today I am going to deal with monohybrid cross. Mendel's monohybrid cross. For doing the monohybrid cross, Mendel selected true breeding tall plant and true breeding dwarf plant. Here, what is true breeding? True breeding means it is a pure line. That all description and what the idea about true breeding pure line is always given in the previous class. So for doing monohybrid cross, Mendel selected true breeding tall and another true breeding dwarf plant. So the parents are tall plant and dwarf plant. The tall plant, since it is a true breeding, it can be written as the gene capital T and dwarf plant as small t. True breeding means in any generation, if it is selfed, it will give the same phenotype as that of the parent. If the tall plant is self, it will always give tall plants. All the progenies will be tall. If dwarf plants are self, it will give all the progeny as dwarf. Such line of generation is called pure line. So here the two parents Mendel selected were tall and dwarf plants with capital T and small t, and they are called the first parental generation or the first parent we selected after so many selfing. So it is the pure line which we call as first parental generation. Then what did Mendel do? Then he crossed these selected plants. Progeny he found are all tall. That is the hybrid. All the hybrid he obtained is tall. And that all hybrid he called as F1 generation. That is first filial generation. So in monohybrid cross, once again Mendel selected pure line varieties and he selected two contrasting characters, one parent with a tall character and another parent with a dwarf character. He allowed them to cross, or he cross-pollinated the tall plant with the dwarf plant. In that cross he obtained hybrid, and all the hybrids are tall in nature, which he called as F1 generation. So we can go through the diagrammatic representation of monohybrid cross. The parent already know that tall and dwarf, which tall into dwarf, and since it is the expression we morphologically observe and is called the phenotype, which can be represented as P1. Then the genotype of this tall, since it is pure line, is capital T, and dwarf, which is pure line, we can written it as small t. And this capital T and small t are the genotype. From the tall plant, we get two types of gamete as capital T of one and capital T of another. Since both are the same, we can represent it only in one type. So it has capital T, and from that dwarf two gametes are produced, one with that is small t. Since it is all two gametes are of similar, we again represented it only in one type. And so when we write gametes, we have to put within a circle, or we can write that letter genotype, and there will be, we put a circle around that genotype. So we get that gamete, the circle and the representative letter, we call it as gametes. So gamete must be written as those seen here. When he cross, he get the tall hybrid with the genotype. What is the genotype? That is capital T, small t. And since in the previous slide I say that all the hybrids are tall, again I am saying that these hybrids are the genotype of these hybrids are capital T, small t, that is the F1. Then raising second generation in monohybrid rows. In second generation, the parent he selected are those hybrid he obtained in the first cross. And here tall into tall. That is the phenotype which we can written as the second parent. So how we can represent? We can represent it as P2. And this tall, all the F1 hybrids are capital T, small t, and we can write tall as capital T, small t, and second parent which is tall as capital T, small t. From each parent we get two types of gametes. One is with the capital T and another is with the small t. So we get 4 gametes. And these gametes can be represented as the way which is shown in this slide. And this is the Punnett square table which we can represent the progeny of the F2 generation. Here the male and female sides are represented, which I represented in the first box of the first column and first row as a round and an arrow, and below that there is a plus sign. And usually the mirror with a plus sign is seen in the Greek mythology, as the ladies always look their glamour through a mirror which she put in a coin in her neck, and all time she can take the mirror and look the beauty of her face. For that, from the very earlier time, the ladies are symbolized as with a round and a plus sign. The plus sign that is used as a hand, and here also handle for taking the mirror, but in this here also represented it as a round and a plus sign, and the male it is represented as an arrow which is seen in the first box of the first row and column. And again in the first row we can top of the first row we can write it as the male gametes as capital T, small t, and below we can write on the left row column as capital T, small t gametes. Then crossing each gametes we get capital T in the first green column, which can represent it as tall. Again in the second column, second row, second the rightmost side of the first green row we can write it as capital T, small t, which is also tall. Again the lowest green box as capital T, small t as tall. These 3 green colored column, which why I give these 3 as same color, because these 3 shows same phenotype. And again another with the blue color which is with the small t, and they shows dwarf phenotype. So here 3 tall and 1 dwarf is obtained. So we can write the monohybrid ratio as 2 types of ratio we get. One is the phenotypic ratio and another is the genotypic ratio. Phenotypic ratio is the 3 tall we get as in the green colored box and 1 dwarf in the blue colored box. So as 3 tall is to 1 dwarf as 3 is to 1 is the phenotypic ratio. In the genotype, when we look into the genes or when we look into the genotype, we can notice that there will be 1 capital T, there will be 2 capital T, small t heterozygous, and there is 1 again homozygous, small t. So genetic constituent of this progeny were written in the ratio as 1:2:1, or we can say that it is 1 capital T, 2 capital T small t and again 1 small t, and in short we can say the genotypic ratio as 1:2:1. And from this monohybrid ratio, Mendel constitutes certain postulates from these ratios or from this cross as called postulates based on monohybrid cross. First one is principle of unit character, second one is principle of dominance, and third one is principle of segregation. Principle of unit character, principle of dominance, and principle of segregation. Then we can go into each principles. First one is principle of unit character. Every character of an organism is an independent unit and its inheritance is controlled by a pair of hereditary factors. So in unit principle of unit character, it says that each and every character of an organism is an independent unit. That means it will not, or each character will not depend on another character. So it is an independent unit and its inheritance is controlled by, inheritance means transmission from one generation to another or transmission from parent to offspring. So the inheritance is controlled by a pair of hereditary factor. And what is the factor? During Mendel time he called it as factor, and now what we call it as genes. So once again, what is principle of unit character? Every character of an organism is an independent unit and its inheritance is controlled by a pair of hereditary factors. Then we can look into principle of dominance. When two alternative forms of a hereditary factors are brought into gametic fusion, one of them dominates and expresses in the offspring and suppresses the effect of other factor. What is principle of dominance? It states that when two alternative forms of a hereditary factors are brought into gametic fusion, two alternative forms, we can say it from the example, that is capital T and small t. When these two alternative forms of hereditary factors are brought into gametic fusion, gametic fusion means this capital T can fuse with the capital T can fuse with the small t can fuse with the another small t. One of them dominates and express in the offspring. So in the first generation, capital T is fused with the small t so that one of will be expressed, that is capital T will be expressed. So the plant we observed as a tall and the other is suppressed, or the other factor is suppressed. What is the other factor? Small t is the other factor. So when two factors when brought to fuse each other, this capital T and small t fuse to each other, and what will be the result? One of the character will be expressed, that is the character of the capital T is expressed and the character of the small t is suppressed. So the expressed factor is called dominant and so suppressed one is called recessive. The expressed factor, the capital T, which is as tall, is the dominant. So dominant will be expressed and recessive will be suppressed. Okay. So this is the principle of dominance. Then principle of segregation. And this principle of segregation is called as later as first law of Mendel. Law of segregation has universal application. This law has universal application which says that two alleles of a gene separate or segregate from each other during genetic transmission. That is two alleles of a gene. What are the two alleles of a gene? From the example we cited, capital T and small t. Two alleles of a gene separate or segregate from each other during genetic transmission. During genetic transmission, that is transmission to the offspring, what happens? These factors will get separated and enter into new pairing in the next generation. Enter into new pairing means will be paired with the gamete of the or the allele of the another parent, and it will form a new pairing and get a next generation. We can go through the detail of law of principle of segregation, and this principle of segregation is called purity of gamete. So principle of segregation was later expanded as law of segregation by Correns, and he later called it as the first law of Mendel. In somatic, since it has a universal application, that postulates come into law, so called the first law of Mendel. In somatic cell, hereditary factor exists in pairs. In somatic cell means in vegetative cells there will be hereditary factors in pairs, but in gametes there will be one member of each pair. So in diploid cell there will be two pairs are seen. In diploid cells or in the somatic cell or in the vegetative cell, two pairs are seen for a gene, or hereditary factor exists in pairs. But in gametes, what we only notice, one member of each pair is found. The two factors never blend or fuse together and remain distinct. These two factors, if the capital T and small t was together there, it will never blend. It will be give separately, or it will give separate results. If it blend together, what will happen? We never get a tall, we get an intermediate height plant. Since capital T and small t blend together, what will be the result? It will be the intermediate of the tall and dark. So the Mendel get all the plant as tall in the F1, which proves that these two factors never blend each other when they come together. So two factor never blends or fuse together and remain distinct. During gamete formation, the member of each pair segregate or separate out and distributed to different gametes. During gamete formation, these two alleles again get separated and distributed into different gametes. They come into new pairing during gametic fusion, which Mendel called segregation of hereditary factor. This separation of these two factors into two gametes and coming back together in the next generation, what he called it as segregation of hereditary factors. So students, here I am winding up. Today we deal with how the monohybrid cross has conducted and what is the results of the monohybrid cross and what are the phenotypic and genotypic ratios. We can do these monohybrid cross with the 7 contrasting characters what Mendel selected for his study. Here I can take into consideration only the height of the plant. From the postulates, from these results, Mendel derives 3 postulates as first one is purity of unit characters, principles of unit characters, principles of dominance and principle of segregation. So students, you go through the topic what I taught now and prepare notes and study well. Hope all of you are safe at home. I am winding up. Thank you from Devipriya.